Welcome to Wednesday at the Genome. I'm Stan Fields. I'm a faculty member in the Department of Genome Sciences. This is, I believe, the 10th year that Genome Sciences has been presenting non-technical talks geared for the general public, all of you, to tell you a little bit about exciting developments in genetics, in genomics, in proteomics, in other fields, and University of Washington's part in this research. We'll have, again, 4 talks this summer, and I invite you all to both come to the talks and then to stay afterwards. We'll have coffee, and you're welcome to ask questions and have further discussions with our speakers. Tonight we have Dr. Gail Jarvik, professor and head of the Division of Medical Genetics in the School of Medicine. Gail is from Mount Prospect, Illinois, originally, where her dad was a fireman who set up the first paramedic program in the state of Illinois. So she comes by her medical interests quite early. She was an undergrad and medical student at the University of Iowa, and then she did her PhD at the University of Michigan. And after working at the University of Pennsylvania, Gail came here to the UW as a medical genetics fellow in 1991, and she joined the faculty in 1995, and she became division head, the Division of Medical Genetics, in 2007. And she'll be speaking tonight on genomic medicine. So welcome, Gail.
Thanks a lot, Stan. So brief poll of the audience so I know who my audience is. Raise your hand if you're a student. If you're a student. Yay, students. Raise your hand if you work for the University of Washington in any capacity. Yay, University of Washington. Raise your hand if you are not a scientist or studying science. Okay, all right, this will be fun. I have a talk, and I'm going to give it, but I want people to feel free to interrupt me, to ask questions. This is really an interaction. The goal is to have you learn what you want to learn, and that's what I'm here for. So I'm from a big family. I'm used to being interrupted, so just go for it. Okay. So I'm going to tell you a little bit about medical genetics, which is the division I'm in, and what we do in my lab. And then I'm going to talk about genetic variation in populations and in disease, give you some examples about how we look for genetic variation that might be associated with diseases, and then talk about the future, the precision medicine initiative and data sharing. So that's the general framework. And if there's anything not on that list that you want to talk about, we can do it later. Okay, so medical genetics was founded by Arno Matulski in 1957. And there is Arno right there. And there is Arno again more recently. He still comes to work, 93 years old. And he founded the division as the first division in a Department of Internal Medicine that studied genetic diseases. And then they opened the clinic in 1959. And the clinic tied with Hopkins for having the first genetic services available. So a very forward-thinking man. This is a clinic conference from 1962, back in the old days when they just brought the patients in and cross-examined them in front of everybody. We do it a little differently now. But this is a reunion. We have trainees all over the country who trained in this program. Right now, medical genetics has a clinic that sees over 2,400 adults a year, a lot more people seen on the pediatric side, but this is the adult clinic. Half of that is cancer genetics, breast cancer, colon cancer, ovarian cancer, et cetera. Up and coming, and number 2 is cardiac genetics. So these are people with cardiomyopathies or electrical disorders of the heart, are often genetic, cause sudden death. Neurogenetics, we have a very large neurogenetic service, including autism, Huntington's disease being a common neurogenetic disorder. Connective tissue disorder, we have Peter Byers, one of the world experts on osteogenesis imperfecta, Marfan syndrome, other collagen disorders, so that's a big clinic. And then all-comer adult genetics, and we also train MDs and PhDs in medical genetics. So that's what we do in the program, and what do we do in my lab. One theme of my lab is that we focus on process, which is mathematical genetics. So we try and understand how genes cause disease, and we do math. and we do discovery. And this you can do across diseases. And so we do a lot of different diseases in my lab. So some labs are just like they do this one disease. My lab kind of does everything because it uses the same tools. So we have a big, like 15-year project now on looking at intellectual outcomes in children with congenital heart defects. These are children who have isolated congenital heart disease. They don't have Down syndrome or something else that would affect their brain. Their brain should be normal, but they have a lot of insults around not getting enough oxygen, having inflammatory responses, and they are very variable in their outcome. They have a high rate of behavior disorders and other intellectual issues. Some of them really sail and some of them don't, and so we've been studying what predicts who does what. This is a colon cancer, chopped out of the colon. We do a lot of work on the genetic causes of colon cancer and also getting the genome to the clinic for colon cancer. This is what we call a GWAS, and this is like a whole, this is chromosome 1, 2, 3, 4, 5, all the way to 23. Whole genome, you throw markers across the whole genome and see if any part of the genome is associated with your disease, and this is what a positive looks like. So all these things, these are the p-values over here, and this is where the disease gene occurs for this disease, and this is probably another one. And so that's just another tool that we have for mapping disease. This happens to be for white blood count. We do a lot of heart disease. This is actually the carotid artery. This is right here in your neck, internal and external. You can see plaque right here. If you feel it, you can feel your pulse, but don't feel both at the same time because you'll pass out. It's not a good idea. Many medical students have made that mistake. All right, so inflammatory disease. I do bioethics on the side. I'm not a trained bioethicist. I just dabble because I find it very interesting. And so the bioethics department is very kind to me and put us up with me. And we work on a lot of things together. A lot of medical genetics, pharmacogenetics. How can the gene help you in clinic? And then I still see patients with genetic disorders. So, all right. So the Human Genome Project. I think we've all heard of the Human Genome Project. It was officially 1990 to 2006, although it wasn't completely done in 2006. And the idea was the first draft of the human genome. And for me, I've been in the field longer than this, and I like to do math, and I like to do math with a lot of data, and the Human Genome Project gave me a lot of data to do math with, but it's made a huge difference to our patients. So when I started out as a medical geneticist, I used to frequently be telling people, well, it's inherited in family in an autosomal dominant fashion. Your children have a 50-50 risk of getting it. I can't tell you if they'll get it or not. You know, have a nice day. It wasn't very satisfying, but now I can find the gene, test for the gene, predict it in the children. We can do treatments before people get sick, preventative interventions. So it's a very different world. And that's in thanks to the human genome. So I love this slide, though. When I asked what little girls were made of, I was hoping he would say sugar and spice. But the human genome is essential to all of us. It's made a giant difference for research and for medicine. So what's new? What's really new is what it costs to sequence a genome. So the Human Genome Project, I think they say $300 million for the first genome. So this is really a short span of time to me, but maybe not to the high school students in the room. And this is a log scale, okay? So this is the cost per genome on a log scale, $100 million, dropping down to around $1,000, and actually a little less for the genome itself, a little bit more with the analysis. And so this is really the big change. And this has made genomic medicine accessible for patient care. Because if you're out at this point, you're not going to be able to get that test. But if you're down at this point, it becomes a reasonable thing to do. So this is a headline from Forbes magazine, because they do much better marketing than I do. The first child saved by DNA sequencing. And this is a little boy from Wisconsin, and he had a very severe disease that caused inflammation in his bowels to the point where he had fistulas open between his bowels and his stomach, and was just, sick in bed and literally dying. And they sequenced his genome. They were considering doing a cord blood transplant because they thought it was an immune deficiency, and so that's a way to replace the immune system, but they weren't sure that was the appropriate treatment for him. They did genome sequencing. They found a variant in a known immune system gene. This gave them the certitude to go ahead and do the cord blood, and that cured him. So first patient saved. Now, would they have done the transplant anyway before he died? Probably. But it makes a better story if you say they saved him this way. So, OK. So when did he get sequenced? He got sequenced around here. This is a log scale, like I said, so like $30,000 or $30,000, a lot of money for one test in medicine was done on a research basis. So not everybody is going to get that test, right, at $30,000. But now, maybe so. Still hard to convince the insurance companies to do it, but easier than it used to be. OK, so could have been saved by a genome. This is a patient who ended his life, unfortunately, here at the UW. He was a healthy 35-year-old diabetes. And he started to get short of breath. And he told his doctor about that. And they weren't too worried about it. And they thought he might have asthma. And he goes treated with antibiotics for bronchitis. And then suddenly he couldn't breathe at all. And he went into an outside hospital. At that outside hospital, he had multiple organ failure. So his heart failed, his liver failed, his kidney failed. He was at that point transferred to us, but unfortunately a little too late for us to do anything about it. And they didn't know what was wrong with him. That was fundamentally the problem. It's hard to fix a patient when you don't know what's wrong. For some reason, his heart was not adequately pumping suddenly. No one knew why. Unfortunately, he went to the autopsy service, and he had tumors. And those tumors were secreting hormones that were driving the heart. So they were stimulating the heart. And that was the cause of his disease. And if those tumors had come out, he would have been cured. But no one knew that this was the problem. OK, so the genetic test results found von Hippel-Lindau. If he had had this test, if he had had a genome sequence, because they didn't know what they were looking for, but if they had done the test and found this von Hippel-Lindau pathogenic variant was a known disease-causing variant, they would have known to look at his kidneys, and they could have fixed him. OK, so here's someone whose life could have been saved by a genome if anyone had thought of it and if the tests were available to him. So there really is a promise for even people who don't look genetic at all that this is a test that can help them. OK, a lot of obstacles in the road to get there, though. The patient education needs are great. There aren't a lot of geneticists. There aren't a lot of genetic counselors. The primary care providers also don't know a lot about genetics. I'm going to give you an example of that later. We need to know how these patients and providers, their doctors, nurses, other providers, how they want information, what information is useful to them. Which variants are important to human health is a giant problem that I'm going to delve into a little today. What results do we give back to people? What results don't we? Do we just give people everything and let them decide what's valuable? Should we tell them about disorders that they're going to get that we can't prevent? Should we tell them they're going to get Alzheimer's disease, for example? Do we give patients reports on a piece of paper? Is that the most useful way? How else can we give them reports? How do we get it in the health record? And how do we get insurance to pay for all this? So a lot of questions. OK, so a lot of marketing around genomics. I liked this one. This is lose weight your way. If you get a DNA test, they will tell you the best diet for you. OK? So don't get too excited about this. This is actually a spin-off of a paper or a book that came out a few years ago, like 10 years ago, probably more, Eat Right for Your Blood Type. And it had 4 different diets depending on whether you're type A, type B, A, B, or O. And you know, because there is apparently a blood type where a donut is a good choice. So those are the O's. Yeah, exactly. Thank you, Billy. Yeah. So you know, the promise of genomics, there's a lot of hype around genomics, and not all of it is true. And this one's even better, which is that they're going to DNA type you and find your perfect mate for you. So with regard to diet for DNA, you know, I would say eat a lot of fruits and vegetables and you know, reduce your highly sugared drinks. With regard to finding your perfect mate, you know, I think your mother might have a better chance than this company. So, OK, so what can we do? If we can't do those things, what can we do? Well, Ancestry, we're really good at Ancestry. So a lot of people have sent their DNA off to 23andMe or Ancestry.com and found out where their ancestors are from. That's very accurate. We do love, like, what % Neanderthal are you, which is kind of fun. Pharmacogenetics. For certain drugs, it's very useful to know there are things that can go wrong with drugs. You can need a higher dose. You can need a lower dose. The drug could make you sick as a side effect, or it could not work at all in you. And those are all things that we try and address with pharmacogenetics. What's the best drug for you? And when we talk about precision medicine and targeting the right treatment to the right person, pharmacogenetics is really the low-hanging fruit. So we want the right dose of the right drug and not a drug that will hurt you. Prenatal testing of the fetus via mom's blood. This is a great thing for women who have had an amniocentesis, where we used to stick a needle like into the stomach and get the amniotic fluid, but now we can just do the mom's blood and screen for the baby's chromosomes because some of the fetal cells are circulating in blood. So that's a great advance, speaking as a woman. Carrier testing for couples planning pregnancies. Carrier testing for things like Tay-Sachs has been around for a long time. Used to be done by enzyme level, but now you can do a whole panel of 50 different diseases and see if you're at risk to have children with those common genetic disorders. So we can look and find genetic causes of diseases, even in a gene that's never been seen before. So we can take a patient who we have no idea what they have and potentially find a genetic change by sequencing the whole genome that causes that. One way that we do this is in babies who we think have a new thing that their parents didn't have. And so you can sequence the mom, the dad, and the baby. And if the baby has a change that didn't come from the parents, that is likely to be, but not always, the cause of the disease in the baby. So that even if the gene's never been described before, you can get an idea what's going on. There's much less utility in healthy people unless we're talking about someone with a bad family history. All right, so rare variants are common. So this is just the number of the variants, and these are the allele frequency, and you can only get allele frequency of 0.5 because if you don't have one allele, you have to have another allele. So after 0.5, you're on the other allele. And basically what this is showing you is that most variants are extremely rare. Not very many variants are up here so that many of us would have the exact same variant. There are some, and some of those do cause disease. But for the most part, variants are rare, and we don't know what most of them do, unfortunately.
Well, it actually does say that rare variants are common.
Yeah.
Which it kind of is a little too existential for me to...
Yeah, so if a variant is, any given variant is more likely to be extremely rare than a common variant for that specific variant. So rare variants are more common than common variants, yes. So, scary, I know. Okay, work with me. Okay. Definitions. So sorry, but we have to go through a few definitions. So the American College of Medical Genetics decided that this is how we should divide up whether variants cause disease. And so one of them is pathogenic, i.e. we know it causes disease. We try not to use the word mutation for this. Mutation doesn't mean disease-causing by definition. It simply means a change, right? So a de novo mutation in a baby may or may not cause disease. In fact, most babies have a few de novo mutations that they didn't get from their parents, and mostly they don't cause disease. Likely pathogenic means we think we have some evidence that it causes a 90% chance it causes disease, but we haven't completely convinced ourselves. Okay? Variants of uncertain significance, lots of things live in this category. We just don't know. It's too rare. It hasn't been seen enough times. We don't know if there's a phenotype associated with it or a disease associated with it at all. Likely benign means we don't think it causes disease, often because it's more common. The variant itself is more common than the disease is. So we say, well, it probably can't cause that disease, at least at a high risk, because there's just too many people who have it. And then benign means it really doesn't cause the disease at all. We know it doesn't cause it. Often people use polymorphism for benign, and polymorphism has a specific definition of a more than 1% allele frequency, and things can be at that frequency and cause disease. So we try not to use this either. So we use this. Okay, so next slide. Oh, yeah, so I had to translate this to Chinese for a talk in China, so you have to see it because it was hard. So those poor people had to listen to me with a simultaneous translator. It was painful. Okay. Sorry? Okay. So many variants, so little time. Huge problem in genomics, not just in medicine, but in research, is all these variants, what do they mean? So the average person's whole genome has 3.5 million variants from a reference sequence, 3.5 million for each of us. About 500,000 of those are rare or never seen before. These are the ones we really worry about. The more common things we worry about less. So you'll have about 400 genes that have one of these rare or novel variants. And a lot of those will change the actual coding of the protein. Okay, so this is a lot of change. And I will say the first time I got an exome sequence, which happily wasn't mine, and I recommend that you don't start with your own, I looked through it and I was like, oh my God, this person should have like 10 diseases. Okay, but they don't, because a lot of this variation is tolerated and it doesn't cause the protein to not work appropriately. So a huge amount of variation is just fine, benign, doesn't cause disease. Telling what does cause disease from what doesn't, it can be very difficult, actually. I'm just guessing, I was trying to figure this out from databases, but that there are no more than 10,000 genetic variants that we really know cause a disease. So think about that. Like you have 3.5 million, but in the world, we only know what 10,000 cause a disease. We know a bunch more that don't cause disease, of course. So we need what we call annotations. We need to know, is this variant pathogenic? Likely pathogenic. Is it a US? Is it likely benign or benign? We need to know this. And we're working on it very, very slowly because you need a lot of information per variant. You need to see a lot of people with it to know. So there's a database that we have. This is called ClinVar. And everyone, when you say, oh, I've seen this variant enough times, I have an idea about it, you're supposed to put it in ClinVar. And there are we, and with a bunch of other labs, for putting variants into ClinVar. So the idea is to share this information across the whole country in order that we all can have information to best treat our patients. Unfortunately, some of the labs that are commercial labs are not interested in sharing their data. It's proprietary. They can make money from keeping the information private. And so one of the things we're trying to do is incentivize those labs to share their information. What we've done sort of a backdoor. One of the labs that doesn't share is called Myriad Genetics. And so we all, clinical geneticists across the country, sent all their Myriad reports that they had on patients de-identified to someone who put them all in because Myriad wouldn't do it themselves. There are insurers now that are saying they will not pay a clinical lab if they won't share their information. I like that model. Yeah, good for them. Insurance companies doing something right. Yay. But even still, in this whole database, there are only 136,000 which have clinical assertions, many of which are variants of uncertain significance, of course. So, and if the same variant is entered by two different groups, a quarter of the time they don't agree on what it means. Okay, so we have a ways to go with this. Very important to share this information. Okay, so any questions so far? All right, go.
Okay. Yeah. The number of genome-carrying cells in an individual human is order of magnitude of a trillion?
I haven't counted, but I'll take your word for it.
Which is much larger than the number of humans who have ever lived. So don't you expect that you're going to get a lot of well, mutations, I guess.
So when we're typing people, we're generally typing their blood cells, their white blood cells, right? So there was a great science paper a year or so ago that showed like the more cell divisions that certain tissue has, like the more variants they accumulate, because as you keep dividing, you keep. But we're really looking at germline. And if we're not sure, sometimes we see something in people's blood that we don't think they were born with, we will take a little skin biopsy and compare it to that. Okay? And there are people who are mosaics for important genetic changes, and they may or may not have the disease depending on how many tissues they have it in, and they may or may not pass it on depending on whether it made it into their egg or sperm cells. Okay, but we're really trying to look at what we call germline testing. Tumor testing is a whole other thing, of course. A lot of genetic changes happen in tumors. I saw a question back there somewhere. No? Okay. Yeah, so that's a great question. So in clinical sequencing, we do it a little bit differently than we do in research sequencing. So we do deeper read depth, And also, before we return results to people, for the most part now, we use some different technology to confirm the variant that we see. Sometimes for variants that we know really well, that we know work really well on the technology, we don't bother with that step. But for most novel variants that have not been seen before, we'll do like a Sanger sequence, another technology, to be sure that change is real before we share it with people. So usually if you have a genetic test result, the result is right. Every once in a while I've seen a typo in the report. It is a little, like you'd think that technology would be better worked out. But in general, there was a change. They might have typed a number wrong. But go ahead.
So tell me if I'm wrong about this. I'm presuming that most of these in ClinVar are going to be dominant mutations because, the complement would be there and would presumably be normal. Does that make sense?
I'm not sure I followed the question.
Okay, so ClinVar identifies specific mutations
Yeah.
that are associated with the diseases, but you're only going to see those generally if the complement from the other parent doesn't.
Yeah.
So the complement would be normal, okay, and the ClinVar variant would be dominant, I would assume, or you wouldn't detect it.
So interestingly, it is much easier to show that a recessive disease gene change causes disease than a dominant. Because for a recessive, you're looking for, there are two different broken alleles. And for a dominant, you're looking for one, but there are, like I said, 400 genes that have some rare one thing, but not that many genes that have two rare broken things. So actually, recessives turn out to be easier to solve than dominants. Dominants, you need a lot more family data as a general rule. But it is sort of whatever people are interested in. And so people, like, there are disease-specific databases, and they've put their data over. So like the Marfan syndrome database got moved over to ClinVar, and a lot of different. So it really was sort of like what people were focused on and interested in.
So the 27% disagreement, could that be accounted for by the fact that there's complementary mutations that are seen, in different parts of the genome that they wouldn't see in one experiment, but they would see?
So the 27% is I see a variant and you see a variant, and I think it is pathogenic and you don't think it's pathogenic. So we're looking at exactly the same variant. Sometimes we have different information that we're looking at, though. Sometimes we have insider information the other guy doesn't have, and then they won't match. And that's why it's important to share information. There's actually a project going on for all of those mismatches to open up the information. And when I put something into ClinVar, I need to put in a little paragraph about why I think that. And especially if I have private information, I should be putting that in that little paragraph, because a lot of this will never be published in the literature. So it's incumbent on us to put it there. A lot of the mismatches for ClinVar are older. There's old data in there that are pathogenics, and they're not truly pathogenic. And now that we know, it used to be 10 years ago, we didn't have great information about the frequency of alleles in the population. So if we saw it in a diseased person and then didn't see it in 100 normal people, we'd go like, okay, well, that's interesting. But now we know the allele frequency is for 50, 100,000 people, and we can see if they're too common. And sometimes it's common in a different population. So I have a lot of data on Europeans, but if it's incredibly common in people of African ancestry and they don't get the disease, it probably doesn't cause disease too. So a lot of these older interpretations turn out to be wrong, and so we don't use old interpretations even from ClinVar.
Well, I thought it might have been from another gene that has a mutation in some populations, but if you have gene A and then gene B, and they complement each other and cause the disease, then you're likely to detect it as gene A. But if in another population it's C that
I see what you're getting at. Yeah, so ClinVar doesn't even deal at all with gene-by-gene interactions. It's simply highly penetrant Mendelian one-locus diseases. Yeah, it gets harder. And we don't even have good nomenclature for that. We have a long way to go when multiple genes are involved. Yeah. Okay, other stuff. All right. Okay, so I'm going to give you an example of how we looked at a bunch of genomes to see how likely it is in relatively healthy people that you're going to find something interesting. So high penetrance, that means if you get the genetic change, you're very likely to get the disease. disease-causing, hopefully intuitive. Rare, so this is my example that the rarer variants are the ones that are more likely to cause high penetrance disease, because if it caused disease at a high rate, it would get reduced in the population over time. Actionable. 2 reasons to say actionable. Like I said, if you can't fix it, I tend not to tell people about it. But also, people have this idea that if you have a genetic change that causes a disease, that you're stuck with that you're just stuck with the disease. And that's just not true. And so this is my opportunity to say, think about cholesterol disorders. If you have a gene that causes you to have high cholesterol, for the most part, the cholesterol-lowering medications work for you. Okay? So just because it's genetic doesn't mean you can't fix it. And so for those of us who, say like, well, my mom's overweight, so I'm just stuck and there's nothing I can do, well, probably, is something you can do. So genetic doesn't mean unsolvable and unfixable. And that's really important for what we do, because we believe by finding these disorders, the risk for these disorders before they happen, we can intervene to protect people's health, or there'd be no point. Okay. So again, what are you going to find in a relatively healthy person? All right, so what we did is we had a group of incredibly smart people. There were like 25 of us. I think we added it up, and there were like 400 or 500 person-years of genetics experience in the room. 60 of it was Arno, but there are some more. There are some more of us as well. And we really had a very diverse group of people across all areas of genetics, but also an ethicist and some lab people in the room as well. And what we did is we came up with a list of like 112 genes that if you had a disease-causing genetic change, you could do something about it to protect your health. That's what we were going for. So then we looked in 6,500 people's exomes for changes in those 112 genes. And this is what we found. So 4,300 of these people were European ancestry, and 2,200 were African ancestry. And pathogenic changes that we know cause disease, they were known in the population, less than 1% of people and fewer in the African ancestry population. Likely pathogenic around 1%. And these are novel. We didn't know about them before, but they broke the gene, so we think they would cause disease. A few of those, more over here. So really, like less than 2% of people are we actually finding some genetic thing we weren't looking for in them. And so when these companies are marketing, like, get your genome sequenced and protect your health, that's a statistic to keep in mind. 2% of people will have something that really is highly actionable, make a difference. Why is it lower in African ancestry people? Any guesses? Yeah.
I had an earlier
Okay, go earlier question.
Is that 2% number likely to increase over time as you identify more things?
Yeah, so that's a great question. And it's not, my opinion, it's not going to increase very much. It'll increase a little because copy number variants weren't in here, and so there'll be a little bit of that. But it's going to stay under 4%, in my opinion, because the diseases themselves are not that common. When does it get higher? It gets higher when we start to understand what the question over here was about, gene-by-gene interactions, things that double your risk, triple your risk, but don't take you this far. Sorry?
Or when treatments become available for specific things.
Well, we can hope for that.
Right?
Yeah, sure.
And does that include any of the replicated sections? I understand there's like transcription errors that will replicate sections. Does that include any of that?
So no, it does not include that. We call those copy number variants, and that they're not included. Yeah.
Oh, that's what CNV is.
Yeah, that's what CNV means. And then, good for the caveats. Yeah, so we don't have copy number variants in there, sorry. So I tried to translate this into English, but it didn't succeed completely. And then this is a really important caveat, too. In Ashkenazi Jewish ancestry people, breast cancer pathogenic changes alone, there are 3 founder changes, and 2.5% of Ashkenazi people, so 1 in 40 Ashkenazi people, will have just one of those changes. And so that's a very different population that's going to have a higher rate. Okay, so back to the African ancestry people. Do African ancestry people have fewer disease-causing variants? No. What they don't have is good literature. We don't know about their variants in disease because it's an understudied population. Okay, so health disparity there. Okay, and that's why the novel expected disrupted are higher, because these should have been pushed up here because they should have been seen years ago, but they weren't because no one studied it. Okay, so what were they? Sorry, this is really small. But the number one things that we saw were, not surprising to Mary Claire King here, breast cancer, BRCA1 and 2. And so that is by far the highest yield test in the genome. And of course, BRCA1 and 2 cause breast and ovarian cancer at a high rate, pancreatic cancer and prostate cancer at a lower rate. These could have been in men, absolutely. Colon cancer is next on the list. Colon cancer genes, very important, sort of tied with high cholesterol and cardiomyopathy. And then malignant hyperthermia is this thing where when you have a certain kind of anesthesia, you get a very high fever. And if that happens to you in a hospital, they can take care of you. But if it happens to you in the plastic surgeon's office, it doesn't always go well. And then some rarer things. But these are the big players. And this is really what we see in clinic, too. Breast cancer, ovarian cancer, colon cancer, cholesterol, cardiomyopathy. Okay, so those are the things that you're going to find for the most part. And then some other things less commonly. But those are the big players right now for high risk of causing disease. So when do I see a geneticist if I'm worried about this? Breast cancer. Premenopausal breast cancer in both breasts separately, not metastasized. Breast cancer in a male, multiple affected relatives. If you have breast cancer and family members with ovarian and pancreatic cancer, frankly, everyone with ovarian cancer should be tested for this. Oh, yeah, it's here. And then if you're Ashkenazi Jewish, come see a geneticist if you have breast cancer. Colon cancer, early onset, multiple affected relatives. If you also have uterine and ovarian cancer, that can hang with colon cancer and some genetic disorders. Or if you have lots of polyps, come see a geneticist. So, Angelina Jolie. You know, I'm not very good on like stars and stuff, but I give her credit because she has a BRCA, I don't know if it's a 1 or a 2, BRCA1 pathogenic variant, and she's made a big public deal about it and about women needing to get tested. And it's really a service because a lot of people don't talk about this. So a lot of people got tested in the wake of her getting tested. All right. So questions so far? Yeah.
So for that 2% number, there weren't any neurogenetics or anything?
These were only actionable things. So if we can't prevent them, treat them, modify them, they're not there. And unfortunately, a lot of neurogenetic disorders are not modifiable. Yeah.
So ApoE4 was not on that?
ApoE4 is not there. No Alzheimer's genes there. Other questions? Good questions. Okay. All right. So how do we find genes for diseases or other phenotypes? So I'm using the word phenotype because disease means it's bad for you, and I'm going to give you an example of something that's not necessarily bad for you, but it's a good example. And we're going to talk about both high and low penetrance, finding genes that are associated with both high and low penetrance phenotypes. Okay, so we're going to start with this phenotype, and this is why I'm not calling it a disease. So this woman came into clinic and saw Dr. Fuki-sama, who's here. Fuki, raise your hand so we can all acknowledge your brilliant pickup in this case. And she came in for an unrelated reason, but then she said to Fuki, you know, my whole family is strong. This is pretty strong. And we don't really work out. And what's that about? And so, I mean, we'd all love this problem, right? So this is her in one of these fitness competitions. And she really does swear that she did not really work out. And she's pretty cut. And this is her again. And this is her. So this is her in her 20s, and this is her around 40. So it's not as striking at 40 as it is at 20, right? But it's still, you know, the picture's not great, but she's still very muscular. So that was the proband, we call her, because the first person in the family, we call it proband. Okay, so we're calling this the buff gene. And there are known genes for similar conditions of very hypermuscular people. And the first thing we did was sequence those, and she didn't have any of those. We collected the family, so this is the family, this is her, and these are her children. She had children by 2 husbands, which works out great for genetics, by the way. These are identical twins, not useful for genetics, we only need one. So, you know, so she had an extra one there. And then 2 daughters over here. She has a brother who his friends called him the Incredible Hulk when he was a younger man. And her brother also was written up in the newspaper because a small plane crashed and he lifted it up so the people could get out. They believe his daughter is affected, the mother and the grandfather. And the grandfather worked on the railroad and reportedly would carry 2 railroad ties when normally it takes 2 men to carry 1 railroad tie. And still at like 83, though, is healthy and chopping wood. So one of the things you would worry about with muscle hypertrophy is, do you have cardiac hypertrophy that can kill you at some point? But he's doing fine at 83. So overall, seems like a great trait in the family. Are they shorter than average? Because she's 4'10", 5'6". Hard to know because they keep marrying short people, right? So we call that assortative mating, and so it's hard to separate out the shortness from anything else. Because if you are short and you marry someone short, your children will be short. So take it from me, I'm a geneticist.
But Granddad also had kids at 20.
Yeah. Well, I mean, in that generation, it was not unusual to have kids at 20, right?
Okay.
You know, now it's young, but then it was old. So, okay, so these are the boys. So this is them at rest, and that's them flexing, and that's one of their legs. And again, they say they don't work out. So someone I worked with wanted to make a poster out of this, but there are only 17, so I said no. This is the 11-year-old, and it's, again, hard to appreciate, but she is really a muscular abdomen. And this is the baby doing a hang, which I love. And the baby also very muscular. So, okay, so how do we approach trying to figure out what they have? They had no known genetic disorder that does this. That we did that early. And so this is the strategy that we had. And we chip the whole family, we call it. We get genetic markers from the whole family, and we say, the people who have this trait, what do they share in common? What part of their whole genome are they sharing? And then we're going to sequence through that little part and see what the change is. Okay, so that's what we did. We used tools that Brian Browning, a brilliant mathematician, made. As I said, Fuki found the family and collected the whole family and got all the phenotype information for us. And we went ahead and did this. So there's the family again. We sequenced and tested a bunch of people. It was a sensitive thing in the family. We ended up not collecting any of the people who don't have it because they're sensitive about it. Okay, so the program did not want us to talk to these siblings because they are not happy to not have this trait. Okay, so you can understand. Okay, so when we mapped it, the only thing shared among all the affecteds was this bit of chromosome 17, and it looks like 2 bits of chromosome 17, but probably there's an error in the sequence or something here. So this is probably 1 big bit of chromosome 17. So that's what it looks like on chromosome 17. And you go like, well, that looks small. You should be able to figure that out. But all of these genes, all of these little names are a gene in this region. Okay, so a lot of genes still. All right, and that's common in a single family. You don't have good crossovers. You get very giant genetic regions. And so this is where low-cost sequencing is really helpful. So we sequenced through that region and a bunch of people in the family. And this is what they shared. They shared a change in this gene, SCN4A. It is relatively conserved in evolutionary time, although not terribly. This is the amino acid substitution in the protein. And this is a skeletal muscle gene, which was good. That's what we wanted. We thought it would be in skeletal muscle. And it's a sodium channel. And it's known to be associated with a couple other diseases. One of them is called hyperkalemic periodic paralysis. And these are people who have high potassium, hyperkalemic. And then they have, like, freeze up. Well, our family didn't have that symptom at all. So one of the things that we've learned in the genomic age is you don't know what a gene does. And so I've definitely been in meetings where we're looking at genes in a certain region, and someone says, well, it can't be that gene because it does this. And we say, no, you don't know. That gene could do 2 other totally different diseases. And that, I think, has been a real lesson. But there is another genetic disorder associated with this called myotonia congenita. And these are babies that are born, and myotonia is like spasms of the muscles. And those babies have actually very bulky muscles because they're constantly spasming, which basically builds them up. Okay, so we figured this, that we think this is it. It's really hard, though, because we haven't found another family with this. So we'd love to find another family and see that they have exactly the same genetic change. That would make me feel better, right? Because I don't know for a fact that this is it in this family.
Yeah, what does missense mean?
Missense means that, that the change causes a change in the protein sequence. Okay?
With as many people as are sequenced and on record, couldn't you go searching for that change
What
in the record?
Yeah. So I actually have other people with this change. I just don't know if they're muscular or not. So annoying. Because it doesn't say in your health record if you're, like, healthy,
But
right?
You can't translate it back and go ask.
So it's really hard to get a grant to collect healthy people to see if they're muscular, it turns out. I mean, really, like, this is something we've thought about. Like, how do we go back and actually phenotype these people? And then a lot of the people I study are older, and you really want people in their 20s to really see this. So so there is probably a way to solve it. We just haven't gotten there. But there is one thing that made us feel better, is that it turns out that there's a horse model for this. So this is a horse who has high muscularity, and this horse is known, these are quarter horses, I guess, because I'm not a horse person, but if you are a horse person, you can correct me, that this was actually selected for in these horses for years because these were these very muscular horses until they started having periodic paralysis, a few of them. And then they said, oh, gee, even though most of them were fine and healthy and muscular, some of them had paralysis, and they would just fall over. That's what happens when you're paralyzed. And that turned out not to be a good thing in a horse. And so they don't select for it anymore. But it's the same gene, and it seems like a very similar trait. So we would call this like an animal model. So that was very reassuring. And what we think is happening is at a very fine level, the individual muscle cells are depolarizing and contracting and building themselves up. And why that phenotype attenuates with age, it gets milder with age, I can't really say. But all of muscle, unfortunately, attenuates with age, as the older people in the audience can assure you. So anyway, so you can do the same thing with disease traits, and we've done it here. This is 2 cholesterol traits, a phospholipid and triglycerides. We did exactly the same thing, exactly in families, just not as pretty pictures in cholesterol as there are in muscular people. So, okay, so that's how you find like rare disease traits. And that's something we do a lot here, and not just in my lab, but across the whole Miracle King's lab. There's a whole program called the Center for Mendelian Genetics here, which is trying to solve phenotypes that they collect from all over the world. All right, moving on. So I said before, common variants don't make big health impacts. That's not to say they don't have any health impact, but they don't make big health impacts because they wouldn't stay in the population. So I'm going to give you an example of common variants with a health impact, and I'm going to use Neanderthal. So this was a consortium effort that we were involved in, and it was led by UW and Vanderbilt, but used data from 9 sites that had health records on people that were very deep, a lot of information about people's health, and then also genetic data. And we combined it, and they know from studying Neanderthals which genetic variants come from Neanderthals that are in current human populations. So we looked at those variants and tried to associate them with human diseases. And there were about 28,000 people in the final study. Okay, so Neanderthals, as I think most of you know, interbred with humans, and then they died out. And about 1% of Asian and European genomes are of Neanderthal origin, varies a little. So why do we still have Neanderthal variations? Why didn't these get sort of kicked out over time? Maybe they were useful for something to our ancestors. Okay, so we went ahead and we looked for association with disease, and this is what we found. So these are different diagnoses. Actinic keratoses are sort of the thickened plaques that older people get from the sun. Mood disorders, depression, obesity, another skin finding, seborrhea, weight, respiratory infections. And these are the ones that held up the best, actinic keratosis and mood disorders. But what you can see is they explain, for the most part, less than 1% of the trait. So even though these variants are associated with disease, the risk isn't very high. They're not really, they don't have much to do with it. Why? Because these are very complex traits. Lots and lots of genes affect weight, affect depression, affect skin. We also replicated, these were actually already in the literature, so hypercoagulable, protein calorie malnutrition, urinary infections, and tobacco use. Okay, so first, can someone tell me why we only found associations with bad things? Why did the Neanderthal not give us any nice, good legacy of happy things? Yeah?
Because they used to be good.
Well, I think they used to be good. I totally agree with you. But, yeah, that's right. But we didn't look at good things. We didn't have in the database you're good-looking, you're attractive, you're a long-distance runner, because we're comparing these with medical records. So everything we looked at was a disease. So we can only find associations with diseases. So we don't know what positive things any of this caused. But let's go to your point, because you made an important point. So why did they have these variants? Well, probably they were good then, and probably they helped our ancestors. Okay, so...
Is there any proof that they're now bad?
Well, they're associated with these diseases, okay? But they may do good things as well that we don't know about, right? Because we just didn't test that, absolutely. So Neanderthal did not have tobacco, right? So they didn't have a genetic variant for tobacco smoking behavior. That's not why they have it. They must have had it for some other reason, right? It may be some positive associated trait that we don't know about, but it wasn't because it made them use tobacco, because they didn't have it. Okay, rapid clotting, hypercoagulation, may have been really useful when you didn't have doctors to sew up your wounds. Okay, so that may have been a positive, and in this case now it's associated with deep vein thromboses and some other things. But we don't bleed to death as much as the Neanderthal used to. Aggressive inflammatory response, again, in the pre-antibiotic era, you needed to knock those infections out by yourself. Now that can be causing downstream allergies and autoimmune disease, but in the past could have been useful. And the same thing, protein calorie malnutrition. Well, we don't know what they had in their diet. It may have been very different diet and weight regulation. A gene for obesity now might have been what kept you alive 50,000 years ago, keeping enough of those calories. So very different environment. Okay, but the overall point really is how tiny the effect sizes are for common variants in complex disease. So that in order to understand, what is my risk of heart disease, unless you have, something that raises your cholesterol by hundreds of milligrams, you're adding up lots and lots of different genetic effects. And it becomes very hard modeling to figure out what the risk is. Okay, questions about that? Common disease, little effect size. Okay. All right, so precision medicine. So this is the future. President Obama, in his State of the Union address, not this year, but last year, announced this precision medicine initiative of collecting a cohort of at least 1 million people, that they're going to get tons of medical, biometric, blood work, and also genetic data on. It was the only point of that State of the Union address where both sides of the aisle got up and applauded. So yay for precision medicine bringing the country together. I think that when they did the program rollout, he borrowed this from NIH, I'm told, to have the DNA model in the thing. So it takes a huge amount of data to understand what these variants do and how they interact with each other and how they interact with exercise and with diet. And so this will be a start at doing this. And the University of Washington went in to be a site for precision medicine. We are not out of the running yet, but they haven't picked the winners yet. So we're anxiously waiting on pins and needles to see if you too can enroll to be in the precision medicine cohort. So that brings me to kind of my last point, which is sort of the social part of genomics. So we live in now what people have heard this phrase, the sharing economy. So sharing economy is peer-to-peer. It's your Airbnb and your Uber of moving things down a level. And we're doing that in genomics as well. And there are very good models in genomics of taking rare variants and making a series of connections so that someone else who has that rare variant, the exact same variant in another country, can find you, and you can put your information together. And one of those sites is hosted here in Genome Sciences. Mike Bamshad developed this with Debbie Nickerson, MyGene2, for sharing of rare variants and potentially disease-causing variants. So if I have a patient And actually our hypermuscular family, that variant and the phenotype are in one of these sharing systems, and so far I haven't hit on anything, and so are some of the other variants we have for other rare diseases. And so people will contact you and say, like, oh, I have that variant too, or I have a different variant in that same gene. What's your phenotype? What's my phenotype? But what we're trying to do, I'm not sure I'm part of that we, but what many people are trying to do is get people to share their genetic data broadly. So a cancer patient would get their raw data back, and then they could upload it into some database, and that would be shared with everything. And if you've heard about like the cancer moonshot, that's cancer genomic data, but the idea is really this incredibly broad data sharing. So from my standpoint, it remains to be seen whether someone who has cancer is motivated to share their whole genome publicly with a lot of information about themselves. I don't know how that's going to work, but that's a model people are talking about. So it'll be interesting. One of the things that what we're proposing to do in a grant that we'll see if we get is to give people their raw data back and give them like a list of here's places you can do it and then see what they do, because nobody's ever asked that question. So how many people here would share if you had your genome sequence, would you share it publicly? Yeah, that's good for you. I would. How many of the geneticists would? No, just kidding. Okay. So I think I'm going to skip over that and just say, in summary then, lots of variants, a lot of work to be done. Don't run out and get your genome sequenced now, but if you want to know your ancestry, go right ahead. And the cost of sequencing is still coming down, and then what we know about sequence, how we interpret it, is going up. So I think the sweet spot is coming very soon. Precision medicine is going to require a lot of data and data sharing. And then really, I'm up here talking about a lot of people's work. These are sort of the closest members of my team, but there's a lot of other people whose work is represented here. And then the Genome Institute and the Cancer Institute funded a lot of the research that I talked about today. OK, so we'll take more questions.